very good morning. I hope and I believe all of you are doing well. So just 2 more days to go. Let's say 3 more days. We have 15, 16, 17 for our exam. And I'm sure all of you have your last minute plans ready. You know what you want to revise in this last 2 to 3 days. Please focus on that and do not go too much into what to read, what not to read, that sort of stuff. Be very, very sorted and clear that what you want to revise in the last 2 to 3 days. Now, since yesterday in the infectious diseases, we had a few questions on bacterial toxins and it was a bit confusing topic for many of us. So we will quickly summarize and revise this topic of bacterial toxins. Before that, a quick note whether the audio visual is all good. Right. So the plan for the today is after this session, then we would have 5 p.m. and your 10:30 p.m. the final sessions in the series of daily 5 p.m. and 10:30 p.m. sessions that we had. 5 p.m. it's gonna be a mixed bag mega live quiz. So the final assessment for your preparation, you can say that. And 10:30 p.m. it's going to be your predominantly ortho ka revision. Radio, I have already taken top 50 images. I hope all of you have seen that. If not, at least watch it before your exam once. It's there on the Unacademy app in the special classes. Today orthopedics, named fractures, splints, braces, bone tumors and your nerve integrity test. That is what we are going to see in orthopedics. Extremely important. Yes, so today the 5 p.m. 10:30 p.m. sessions are the last ones. These 10 a.m. sessions, maybe 1, 2 we will have in the last days, but not really sure. Let's see how it goes and then we will decide. All right. So let us start with the session here. First, to differentiate between exotoxin and endotoxin. We should first understand the difference between exo and endotoxin. So exotoxin is present in your gram positive and some gram negative bacteria. Endotoxin, remember endo is N. It is present in the outer cell membrane. Very important. Where is it present? In the outer cell membrane of the most gram negative bacteria. So N for N, endo is gram negative. We know 1 gram positive bacteria which has endotoxin. Janani, I have discussed IPC in the Unacademy Plus platform in detail, all the IPCs. In the recent special class of FMT, I have discussed few there as well. Gram positive organism which produces your endotoxin is Listeria, right? It's Listeria. Listeria we know important in your meningitis, in your neonatal meningitis, tumbling motility, वो सब हमने देखा है. Now, exotoxin, as the term says, it will be secreted out. It is exotoxin. So, toxin goes out of the bacteria. So, is it secreted from the cell? Yes, it is secreted. Endotoxin is not secreted. The bacteria has to undergo lysis. The cell membrane has to undergo lysis to have that action. These exotoxins are predominantly polypeptides. While in your endotoxins, you have multiple components there. The active component is your lipid A component. Remember lipid A is active of the lipopolysaccharide. Okay, so it is the structural part of the bacteria, the lipid. That is why it is released only when the bacteria is lysed. The location of the genes, it is either from plasmid or bacteriophage. We know the mnemonic A, B, C, D's, the bacteriophage wala bacteria. Now this is your bacterial chromosome. Adverse effects are very high with exotoxin. They are very high adverse effects, and the endotoxin has low effects. We will see now for the exotoxins. Endotoxins, remember, they basically induce your TNF, interleukin 1, interleukin 6, all your pro-inflammatory cytokines, right? Your pro-inflammatory cytokines. So they cause fever, shock and DIC like you have in your Neisseria, gram-negative organism. It causes DIC because it has endotoxin, TNF, interleukin 1 and interleukin 6. Exotoxin is very antigenic and they can induce the antibodies, that is antitoxin. Endotoxin is poorly antigenic. So we have tetanus toxoid, right? Tetanus, Clostridium tetani has an exotoxin. So toxoids are used as vaccines. Here you have no toxoids formed and there is no vaccine available. Heat stable, your endotoxin are stable at 100 degrees centigrade for 1 hour. That means they are heat stable. These are destroyed rapidly at 60 degrees centigrade. Exceptions being your staphylococcal enterotoxin and the E. coli heat stable toxin. The heat stable toxin. Anyone can tell me what is the mechanism of action of heat stable E. coli heat stable toxin? Is it cyclic AMP? Is it cyclic GMP? Is it your protein synthesis? What is the mechanism of action? Stable, we will see it acts via your cyclic GMP. Okay, it acts via your cyclic GMP. Heat labile acts via cyclic AMP. So it is cyclic GMP. Alright. So diseases, exotoxin, tetanus, botulism, right, spastic paralysis, flaccid paralysis, diphtheria, elongation factor 2, cholera, cyclic AMP, all these are your exotoxins. Meningococcemia, which causes your DIC, gram-negative rod sepsis, these are your endotoxins. Okay, so the term itself tells you exo matlab bahar secrete hone wala, endo matlab staying inside. Only when the bacteria is lysed, it will have that effect. The active component is lipid A, it is heat stable, it is poorly antigenic, it causes pro-inflammatory cytokines, fever and shock. Okay, now this is your image showing the exotoxin. Look at how the exotoxin is secreted out without the lysis of the bacteria. Exotoxin acts on the receptors on the host cell and then it causes the downstream cellular reaction like increasing the cyclic AMP, cyclic GMP or elongation factor 2. While your endotoxin, this is the endotoxin which you see, only when the bacteria gets lysed, the endotoxin is released and it leads to your pro-inflammatory cytokines leading to your fever, shock, okay, fever, shock, sepsis, that is what we see. Alright. Uske baad, let us see the mechanism of action of various toxins. The group A is your inhibiting protein synthesis. Which are the bacteria toxins acting by inhibiting protein synthesis? इसमें हमने one mnemonic we have already seen, Corynebacteria diphtheria CDEF. It inactivates your elongation factor 2. We saw this in our biochemistry class also. So it inhibits your protein synthesis. So your diphtheria toxin is by inhibiting protein synthesis. Similarly, remember Corynebacterium diphtheria di and Pseudomonas do. So ye dono, di and do, they basically act via elongation factor 2. The exotoxin A of Pseudomonas also acts via this mechanism. The other manifestations with Corynebacterium diphtheria, very, very important. There is pseudomembrane in the throat, grayish white, and there is bull neck. Remember bull neck, very, very important. And there will be myocarditis. Okay, there would be myocarditis. Shigella toxin, Shiga is your 60. Okay, so remember it that Shiga is 60. Okay, Shiga is 60. That means it inactivates your 60S ribosome by removing the adenine from rRNA. And similar to Shiga, we have Shiga-like toxin, which is seen with EHEC, enterohemorrhagic. Shigella, EHEC, they basically cause your hemolytic uremic syndrome, hemorrhagic, hemolytic. So they both have the same mechanism. Shigella, by causing the GI mucosal damage, leads to dysentery. It also enhances cytokine release and causes hemolytic uremic syndrome. Shiga-like toxin also causes cytokine release. It causes your hemolytic uremic syndrome, classically O157H7. And remember that unlike Shigella, the EHEC does not invade the host cells. Okay, it does not invade the host cells, your EHEC. So protein synthesis wale ho gaye, either it is your elongation factor 2 or it is your 60S. Elongation factor 2, Corynebacterium diphtheria and Pseudomonas. 60S is your Shiga and the Shiga-like toxin of EHEC. This is your protein synthesis. Which are the ones which increase the fluid secretion? So increase the fluid secretion is basically by increasing the cyclic AMP, cyclic GMP, cyclic AMP or cyclic AMP. So basically whatever increases cyclic AMP or cyclic GMP will increase your fluid secretion. Now E-Tech, which causes your traveler's diarrhea, right? T for T, enterotoxigenic. E-Tech causes your traveler's diarrhea. It has a heat labile toxin, it has a heat stable toxin. The mnemonic to remember is labile in the air, stable on the ground. So it is labile in the air and stable on the ground. Air matlab adenylate. So labile wala is AMP. Stable on the ground, that is stable is by the GMP. So labile in the air means cyclic AMP. Stable on the ground means G, that is cyclic GMP. So stable wala acts via cyclic GMP. That is what I asked you just now. So both of them, they overactivate the respective cyclase enzyme, adenylate or guanylate cyclase. So increase in cyclic AMP increases the chloride secretion in the gut, and your increase in cyclic GMP basically decreases the reabsorption of sodium chloride and water in the gut. एक secretion बढ़ाता है, एक reabsorption कम करता है. Basically both of them are increasing the fluid in the gut. So that is how they cause fluid, that is your watery diarrhea. Okay, it causes watery diarrhea. Bacillus anthracis, you can remember it the best by our yesterday's clinical scenario MCQ we had in the 5 p.m. class. What was that question? A patient who had gone to a zoo 5 days later developed a papule which then ulcerated, and there was edema in the surrounding tissues. The question was, this edema producing toxin is similar to what? And we said that it is your cyclic AMP. So remember, anthracis is your AMP, Bacillus anthracis, anthrax toxin. It mimics your adenylate cyclase, increases cyclic AMP, likely responsible for the characteristic edematous borders of the black eschar in the cutaneous anthrax. A clinical scenario which we saw yesterday, right? Vibrio cholerae, we read it the most often. Watery diarrhea, the rice water diarrhea classically. Increased water is because of increased cyclic AMP. It permanently activates your GS. Remember, we have seen our mnemonics for GS, GI, GQ. GS increases the cyclic AMP. So it activates your GS, and that is how it increases the chloride secretion in the gut. That is for your cholera. Okay, that is for your cholera. So these are the ones which basically increase your fluid secretion, all of them causing your watery diarrhea, or in the anthrax you have the surrounding edema surrounding that black eschar. These are your increasing the fluid secretion. Stable on the ground, so remember stable is cyclic GMP, rest are cyclic AMP wale. Then the third one is you're inhibiting the phagocytic ability. You have only one here, Bordetella pertussis. So remember P for P, pertussis inhibits the phagocytic ability. Pertussis, it inactivates the inhibitory GS, the GI, inhibitory subunit. That is your GI. So that leads to activation of adenylate cyclase. It increases cyclic AMP. So Bacillus anthracis, Bordetella pertussis, all of them are increasing cyclic AMP. But remember that Bordetella pertussis, it basically inhibits your GI. Okay, it inhibits your GI, and that is how it leads to increase in your cyclic AMP. Cholera increases cyclic AMP by activating the GS. Pertussis increases the cyclic AMP by inhibiting the GI. So remember, it is your inhibiting, that is pertussis is prohibiting. Okay, so you can remember pertussis prohibits the GI, and it leads to whooping cough. There is whoops on inspiration. It can lead to 100-day cough in the adults, 100-day cough. Next one inhibits the release of neurotransmitter. So we have neurotransmitter wala spastic paralysis, flaccid paralysis. So spastic paralysis is seen with your tetanus. Flaccid paralysis is seen with your botulinum. Why will we get spastic paralysis? When you have decreased inhibitory neurotransmitters. that is your GABA and glycine are inhibited. That is why you have increased of activation. So it leads to spastic paralysis, rhizus sardonicus and trismus, that is lockjaw. So this is titanospasmin, botulinum toxin. Both are proteases. They cleave the snare. What is snare? Snare is soluble NSF attachment protein receptor. These are the proteins which are required for neurotransmitter release. Basically, snare proteins are required for neurotransmitter release. These toxins, they break down these snare, the snare protein को break down करते हैं. That is why the neurotransmitter is not released. With your Clostridium tetani, it is your inhibitory neurotransmitter which is inhibited from release, so you get spastic paralysis. In your botulinum, we get flaccid paralysis because the stimulatory acetylcholine is inhibited from release. So it leads to your floppy baby. Very, very important. Botulinum, remember infant botulinum is due to honey. Botulinum ka food poisoning mein history, you will classically have the history of canned food. Okay, canned food is very, very important. Okay, canned food is very, very important. So yes, of course, when you say that the release is inhibited, that means presynaptic, from the synapse, presynaptically you are inhibiting the release. It is not the blockage of the receptors on the postsynaptic. So when they are inhibiting the release, that means, so your release happens from the presynaptic junction, right? आपका release यहाँ से होगा. So both of them are inhibiting the release from the presynaptic terminal. Postsynaptic means the ones which are blocking the receptors, not allowing them to act. So release is presynaptic, right? So this is about your neurotransmitter. Then is your lyses the cell membranes. Which one lies the cell membranes? You have Clostridium perfringens and Streptococcus pyogenes. So the alpha toxin, very important. We all know Clostridium perfringens. It leads to gas gangrene, myonecrosis because it lyses the cell membrane. Why? Because it is a phospholipase. Phospholipase matlab lecithinase. So it degrades the tissue and the cell membranes. So the degradation of phospholipids, basically the cell membrane, leads to myonecrosis and it leads to hemolysis as well. So that is why remember that Clostridium on blood agar will give you the double zone hemolysis. Okay, there would be double zone hemolysis. Streptococcus ka streptolysin O, right? Streptolysin. So it is lysing. So it lyses the RBCs and that is how it contributes to beta hemolysis, the complete hemolysis. Host antibodies against the toxin. We have the ASO titer, anti-streptolysin O. They are used to diagnose rheumatic fever, right? ASO titers help in diagnosing rheumatic fever in your Streptococcus pyogenes. Remember, it is Streptococcus pyogenes, streptolysin O. So which are the ones which lyse? It is your streptolysin O, and your alpha toxin. Please remember, it is alpha, not beta, gamma, delta toxin of your Clostridium perfringens. So remember, perfringens perforates the cell membrane. Perfringens perforates, streptolysin O is also leading to, streptolysin O is also leading to the lysis. Okay, so both of them, they lyse the cell membrane. So we have seen 5 groups of action so far. A quick revision. Protein synthesis inhibitor, EF2 या 60S. Let us revise the toxins that we have seen so far. So we have seen the 5 groups here basically. Right? So A wala, inhibiting the protein synthesis. Inhibiting the protein synthesis either by elongation factor 2 or via 60S. Elongation factor 2, CDEF, Corynebacterium diphtheria, and your Pseudomonas. 60X is your Shiga toxin and your Shiga-like toxin. Which is the Shiga-like toxin? Shiga causes dysentery, hemorrhagic wala. So remember, it is your enterohemorrhagic E. coli, EHEC, which causes hemolytic uremic syndrome 157-7, right? Uske baad, what is the next group that we have seen? Which increases the fluid secretion. Either it is via cyclic AMP or it is cyclic GMP. Stable on the ground, so it is your heat stable toxin of enterotoxigenic. Your cyclic AMP is your heat labile in the air. Cyclic AMP also is your Vibrio cholera. It basically stimulates your GS, increases cyclic AMP. Other one, which is the anthrax, is also via your cyclic AMP. The anthrax toxin is similar to cyclic AMP. That is the edema factor, right? That is your edema factor. और कौन सा देखा हमने? The next one was your phagocytic inhibition. The toxin which inhibits the phagocytosis is your Bordetella pertussis, and Bordetella pertussis acts via inactivating the GI, and that is how it increases your cyclic AMP by inhibiting the GI. Very important. GI wala is pertussis phagocytosis. Next one that we saw, the neurotransmitter release. Neurotransmitter release inhibition inhibits the inhibitory release, that is your GABA and glycine. So therefore it leads to spastic paralysis, is your tetanus, right? That's your tetanus. Inhibits the stimulatory neurotransmitter, that is your acetylcholine. So therefore leads to flaccid paralysis, that is your botulinum, right? That's your botulinum toxin. Next one we saw is which lyses the cell membrane. Which one lyses the cell membrane? Cell membrane lysis is your perforating perfringens and your streptolysin O. Streptolysin O with streptococcus pyogenes, not streptococcus pneumoniae. Perfringens का कौन सा toxin? It is the alpha toxin which has the lecithinase activity. I hope you remember the Naegler's reaction. Lecithinase phospholipase, so it leads to gas gangrene, and remember it leads to double zone of hemolysis on the blood agar. Streptococcus pyogenes ASO titers we do for rheumatic fever, right? It lyses the RBC ka membrane, and that is how it is beta hemolytic. Okay, it gives beta hemolysis. That is what we have seen. Yes, in your neurotransmitter release, basically they act via the snare. The release is inhibited. These are basically your proteases. Okay, these are basically your proteases. All right. Let us go to the next one now. That is your super antigens. Which are the super antigens causing shock? So you have the first two in your microbiology book, the organism Staph aureus and Streptococcus pyogenes. Pyogenes, pus producing wala yaad rakhna, not your pneumonia. So toxic shock syndrome toxin is super antigen. In Streptococcus, it is your erythrogenic exotoxin A. Remember in our yesterday's MCQ, the live quiz, we had the question of scarlet fever. Scarlet fever circumoral pallor tha, that is your erythrogenic exotoxin A. Now toxic shock syndrome toxin, it basically acts via, ye bhi question tha, the polyclonal tampon wala question. Polyclonal T cell activation was the answer. A girl recently started to use tampons, now presenting with fever, altered sensorium shock. That was your toxic shock syndrome. It leads to your overactivation of the T cells, basically both of them. So there is overwhelming release of your interleukins and cytokines that leads to shock. They act via your MHC2. They act via your MHC2. So toxic shock syndrome will have fever, rash, shock. Okay? The other toxins of Staphylococcus, they cause the scalded skin syndrome, which is the exfoliative toxin leading to scalded skin syndrome. And food poisoning is caused by your heat stable enterotoxin, right? We saw in exotoxin, which is heat stable. You have staph ka enterotoxin, which is heat stable. Streptococcus is your erythrogenic exotoxin A. Remember Streptococcus pyogenes, we said RBC lysis bhi karega. Similarly, you have erythrogenic exotoxin A, and it leads to toxic shock-like syndrome, and it can lead to scarlet fever as well. Do you remember for scarlet fever? We saw in the yesterday's pediatrics class. Scarlet fever, you will have sandpaper rash. It is also called as second disease. you will have circumoral pallor, you will have Pastia's lines, right? Pastia lines are seen. How about the tongue? It is white strawberry tongue, then it becomes the red strawberry tongue. First it is white and then it becomes red. So remember, that is basically all red color. You see the rash as well. It is erythrogenic exotoxin A. Okay, it is erythrogenic exotoxin A, which is Streptococcus pyogenes, right? Now coming to endotoxin. Endotoxin we said is present predominantly in your gram-negative, Listeria gram-positive. It is lipopolysaccharide found in the outer membrane, not in the cell wall, outer membrane of the gram-negative bacteria, both cocci like Neisseria and rods as well, like Pseudomonas. So basically it has a O antigen, it has a core polysaccharide and it has lipid A. The toxic component is your lipid A. A is active, that is the toxic component. It is endotoxin, it will be released upon cell lysis or by the living cells by the blebs detaching from the outer surface membrane. So basically the cell has to lyse or there will be blebs formed like this which will release the endotoxin. In contrast to your exotoxin, which is actively secreted. Exotoxin is actively secreted. Three main effects that we see with the endotoxin. They will lead to macrophage activation. Guys, remember the CD marker? ये बहुत important है, very important. Please remember the toll-like receptor. अगर question आता है, this is the most important. Toll-like receptor 4. That is your CD14. Remember monocytes, macrophages. It is 1 and 4. You have CD14, MM. And this is the toll-like receptor which is for your lipopolysaccharide. Very important. Okay. It leads to complement activation and tissue factor activation. Basically, सब कुछ होता है. So yes, Pseudomonas has both endotoxin also and exotoxin also. Exotoxin is the one which we saw, which inhibits the protein synthesis, right? It inhibits the protein synthesis. That is your exotoxin. Endotoxin, ye wala hai, the other one. So the three main effects, macrophage activation. Let us have an, this image. So endotoxin has a lipid A component, which is the active component. It can lead to macrophage activation. TLR4, very important. That is your LPS ka receptor or CD14, which is present on macrophage. Complement activation, tissue factor activation. When the macrophages are activated, you will have interleukin 1, interleukin 6, pyrogens leading to fever. TNF alpha leading to fever and hypotension. Nitric oxide leads to hypertension. So basically endotoxins cause your fever and hypotension. Complement activation, very important. C5 is the one which is responsible for chemotaxis. So you have chemotaxis. C3A is the one which leads to histamine release and you will have your hypotension and edema. When the tissue factor is activated, it activates the coagulation cascade and that is how it leads to DIC. So basically your endotoxin is leading to fever, hypotension, edema, and it is leading to your DIC as well. Very important, remember, complement 5A is chemotaxis, 3A is responsible for histamine release. Okay, it is responsible for histamine release. Alright, so that is for your endotoxin. So your almost all your gram-negative bacteria, like we say your Neisseria meningitidis, when it is causing meningococcemia, that is the one which is responsible for causing DIC, the endotoxin, fever, hypotension, and that is what we see. Apart from that, in meningococcemia, remember an important history, petechiae are also seen, right? DIC hota hai, petechiae bhi rahenge. Now coming to the final, the last slide that we have, basically for action of various bacterial toxins. Increase in the cyclic AMP. I hope you all remember this now. Pertussis. How does Bordetella pertussis act? By inhibiting your GI, and that is how it increases the cyclic AMP. Bacillus anthracis, the edema factor, right? It is the edema factor which is similar to cyclic AMP. So edema surrounding the ischar. Cholera toxin, it causes permanent activation of GS, and that is how it increases your cyclic AMP. Heat labile. Heat labile is labile in the air, stable on the ground, right? Stable is on the ground. So your heat stable toxin of E-Tech, it is stable on the ground, that is your cyclic GMP. Protein synthesis inhibition, diphtheria, di, pseudomonas is di, it is EF2. So diphtheria, di, pseudomonas, though they inhibit your elongation factor 2, and that is how they inhibit the protein synthesis. This is your exotoxin. Okay, this is your exotoxin of pseudomonas, not the endotoxin. Clostridium difficile is also under your protein synthesis inhibition. Remember, unlike your Clostridium tetani or Clostridium botulinum, it is not in your neurotransmitter, it is basically in your protein synthesis. So यहाँ पे भी difficile का die याद रखना, like diphtheria, pseudomonas and difficile. Enterohemorrhagic, that is your Shiga-like toxin, and the Shiga toxin, it is your 60S. Okay, they inhibit the 60S, and that is how they inhibit your protein synthesis. So remember, Shiga is 60. Shigella causes your dysentery, so you will have enterohemorrhagic. It is the enterohemorrhagic E. coli, which is Shiga-like toxin, and it inhibits your 60S. Okay, and it inhibits your 60S. Okay, so yeah, in your toxins you have AB toxin basically. एक A component होता है, एक B component होता है. Remember that the B component is responsible for binding, helping the toxin to bind. A is the one which goes inside, and it is the one which is the active component. So generally what happens in your AB toxin, wherever you have AB toxin, B part helps in binding, A part goes inside, and that is the one which causes the effects that we see. Okay, that is the one which causes the effects that we see. तो यहाँ पे आपका इतना ही दिया है ये वाले में, but you of course know the rest of them. Phagocytosis is your, let me just jot it down here so that you all can revise later as well. So phagocytosis inhibition is your pertussis. यहाँ पे pertussis में याद रखना, it inhibits the phagocytosis. Okay, it inhibits the phagocytosis. Neurotransmitter release is your tetanus, and it is your botulinum. Cell membrane lysis. Cell membrane lysis is your Clostridium perfringens, the alpha toxin, lecithinase, and your Streptococcus pyogenes. Streptococcus pyogenes, the ASO. Which are the super antigens? Staphylococcal ka toxic shock syndrome toxin, and your Streptococcus pyogenes ka erythrogenic toxin A, right? Your erythrogenic toxin is your super antigen. Here you will have a preceding history of impetigo before the super antigen ka effect. Here the associated history is generally the use of tampon. Here it is the impetigo. There will be history of scarlet fever. ये रहेगा आपका super antigen में. Okay. So that is about this session, guys, the toxins. And yes, I will share the PDF on the group, on the Telegram group after this session, so that it's easy for you guys to revise this. Alright, so try solving the questions on your mechanism of action of toxins. I'm sure you have it in your question banks. So please after this session solve those questions so that you are clear on how these questions would be, how the questions can be framed on this topic. Alright. So thank you so much everyone for joining in. That was for this session, a quick revision of toxins, and I'll see you at 5 p.m. with a mega live quiz on the Unacademy app, and then 10:30 p.m. orthopedics ka revision. Alright, so thank you, goodbye.